Good afternoon, and my name is Wes Bickford, and I'm a biologist at the USGS Great Lakes Science Center. I'm based in Ann Arbor, Michigan. And I just want to start off by thanking the organizers of this conference today, because it's been really fun to hear about what's going on in Canada and to think about the potential partnerships in the future. And so I'm going to talk today about some of our ongoing research on cut to drown management, and something that's of a high interest. The Great Lakes water levels are very high. And here we go. All right, so today we've spent a whole lot of time talking about Phragmites management strategies, and I think many of us here on this call know a lot about the different types of strategies for controlling Phragmites. Basically, we can spray it, we can flood it, we can cut it, we can burn it, and all of these strategies kind of have some challenges associated with them in terms of long-term viability, and we've heard some really good success stories on a lot of these, particularly at the Long Point situation. So what I'm going to talk to you today about is kind of combining two of these, the hydrologic management and mechanical management, to cut to drown this plant and effectively kill it. So I think it's useful to go back and look at a historical perspective of Great Lakes water levels. So think about kind of the ways that Phragmites spread and expansion has really tracked with water levels over time. So if we look at this historical graph of Great Lakes water levels, and this is Lake Michigan, Huron, we can see that there was a period of low water around the year 2000 to 2015, and this is highlighted in that box. And this corresponds to a time when the spread of Phragmites really took off. It became really rampant in the Great Lakes. And I'm going to share a couple of aerial images that kind of help visualize this point. And these are going to be images from the Saginaw Bay in Michigan, and this is at the Fish Point State Wildlife Area. And first image is in 1999, and so this, as I said, that period of low water started around 99, 2000, and so you can see here it's kind of difficult to tell with the black and white image, but it looks like water levels are starting to go down, but are still fairly high. But what's really apparent is that there doesn't seem to be a lot of vegetation in the water. It seems to be a pretty open shore situation along this shoreline. Very little vegetation in the water. But as the water levels continue to stay low, if you look ahead to 2005, you start to see plants invading that exposed lake bottom. So when the lake levels are low, it exposes this kind of fertile ground for invasives or other plants to take over, and Phragmites was really good at taking over those areas. So you start to see these green patches forming, kind of the signature of Phragmites, and you can get a sense from this picture just how much the water level's gone down. So you can really see that the bottom of the lake there. Flash forward to 2011, and now we have this like wall of frag across the Saginaw Bay, and I think this is not something that is only happened in Saginaw Bay. I think this is typical to a lot of Great Lakes coastal wetlands, and what we saw when water levels were really low is that Phragmites really was able to expand into these exposed lake bottoms and just take over. And so you can see all these kind of like walking paths that give you a sense for how the water level was low, and it really causes a lot of issues for species diversity and other recreation concerns as well. But as we know, water levels came up, right? So this image is from 2018, and we see a drastic reduction in the Phragmites coverage. And although water levels came up and that helped, this image actually shows an area that was managed also. So in the south here, the landowners in that area took advantage of that high water and also used some chemical treatments to eradicate Phragmites along their shoreline. In the northern region, though, that's state property, and the state did not manage that land. So we have reduced coverage in those Phragmites patches, but the Phragmites still remains. So I just think that's really interesting to show how the fluctuation in lake levels has really allowed for expansion and then even reduction in some areas of these Phragmites patches. And if you're interested in seeing more about this, we put together a geo-narrative that is available online that dives a little deeper into this topic of how variable lake levels have influenced expansion of Phragmites and also management techniques. So I would direct you to the link on the page here, and I can also drop it in the chat after the presentation. I will say that it's a little US centric because the data that we grabbed for water levels was on the US side. So it's a great tool, but also could give some ideas of how it could possibly be expanded into Canadian territories as well. So how does Phragmites survive in high water? Well, it takes advantage of a kind of gas exchange mechanism called convective through flow. And this is fairly common in wetland plants, the ability to be able to get oxygen and other gases down into the root zone and to perform all the vital functions of the plant. So what happens is that air enters in through the stomata of the plant and kind of cycles through. So it'll go down into the root and rhizome system and then efflux out of the dead culms. And so I think that's kind of cool. It's just like constantly cycling gases, which has the effect of oxygenating the rhizosphere, also just giving oxygen like a constant flow of gases to the plant to maintain its functions. Now, as water levels come up, if you look at the image on the right, those processes can still happen, but there's kind of just less surface area for those gases to be able to enter into the plant. So you get a little less through flow, you have more distance for those gases to travel to exit, and therefore it's oxygenating just a little bit less.
Hi, Wes. Sorry to interrupt. Do you mind just speaking up slightly? It sounds like there's just a tiny bit of computer fuzz coming through, perhaps through your microphone.
Okay, I apologize.
No, that's okay. I think I can hear you. It's just a little quiet.
Okay, I will get closer. Sorry about that.
That's okay. Thank you.
So yeah, so Phragmites also has a few other adaptations that allow it to photosynthesize in the water. Gas films can form on the leaves that allow it to maintain photosynthesis even when you have high water levels. We also know that Phragmites plants can quote unquote snorkel through their stems, and so this is kind of the same mechanism that I talked about with this convective through flow, but it comes into play where if the plant is damaged in some way, say through wave action or something like that, where plants break, as long as they maintain the ability to break the plane of the water, they're going to be able to get access to gases and maintain their functions, stay alive, and grow. However, if those plants are broken or are cut below the water level, then we kind of cut off the gas exchange mechanism. And so they don't oxygenate, they are starved of oxygen, and they basically drown. So that's that is, in a nutshell, what we're trying to accomplish with this cut to drown management, is to cut them and cut off the flow of oxygen. So I'm speaking to a crowd that is in a lot of ways more skilled at this technique than we are in the US. This is something that is growing in popularity in the US, but really has been popular for a while in Canada, mainly because of the restrictions around spraying herbicides over water. And so, I know that the Ontario Phragmites Working Group and the Invasive Phragmites Control Center have been doing active cut to drown work. And Janice Gilbert gave a really great presentation on her efforts on cut to drown strategies to the Great Lakes Phragmites Collaborative about a year and a half ago or so. And it was really excellent. And just a funny aside on that, I was talking to some people in the state of Michigan about cut to drown strategies, and they said, "Have you heard of Dr. Janice Gilbert?" And I was like, "Oh yeah, I've met Janice. She's great." And they were just so impressed with the work that she's doing. And she's awesome. We got to get a truck soar, all of these things. So Janice, if you're listening, you have probably a small but devoted following of cut to drown enthusiasts in the US. So I'm not sure if you were aware of that. So even though it's something that's being commonly used, we still have a lot of questions surrounding best management strategies for cut to drown management. And those are, what are the optimal timing for cutting? Are rhizomes actually inviable after they drown? And then what if a clone extends into low water? And what I mean by that is we can have patches of Phragmites that are completely growing in high water, and we can have others that may extend into the shoreline, be connected to the shoreline Phragmites, where if you cut that, it may be able to be rescued through gas exchange from those upland or exposed Phragmite stems. And so we want to understand how that affects the effectiveness of this management strategy as a whole. So we've tackled these questions in a number of ways, but the first one is through a greenhouse study that was conducted last year. And we wanted to understand what the impact of cutting timing and submergence level was on Phragmites survival, and if those rhizomes that appear dead are actually inviable. So we set up an experiment and a factorial arrangement where we varied submergence level and also cutting timing. And we used these mesocosms and cattle tanks in a greenhouse at the University of Michigan. And so just a summary of how it all came together, we collected Phragmites rhizomes from the field, sprouted them in water in a growth chamber, and then planted them in pots, put them into these tanks for submergence and kind of acclimated them to that environment. And to just give you a view of what was going on, you can look at it from a side view. We planted these in topsoil in these pots, and then we placed the pots in these cattle tanks at different levels using just bricks to have the fully submerged plants were sitting on the bottom of the tank, partially submerged, we're about halfway up with some of the plant above the surface of the water, and the no submergence plants were even higher up where that water line was right at the soil surface. This is what it looked like in the tanks. And then as you add water, you can see that some of those plants are poking out just a little bit. Some of them are fully submerged, and some of them are not submerged at all. So in addition to varying submergence level, we also varied cutting. And like I said, we wanted to understand the effect of different cut timing, because managers have suggested that they aren't sure if it's better to do it early in the season or late in the season, or they've seen different effectiveness early or late. So we did an early cut, which is about day 30 after sprouting. We also did a late cut, which was around day 90 after sprouting. And then plant harvest was around day 120. So this can mimic the full growing season. So when we harvested the plants, we just pulled them out of the pots, shook off the soil, separated above and below ground structures, bagged it all, dried it, and weighed it to get an estimate of biomass. In addition, because we were interested in the viability of the rhizomes, we collected some rhizome and looked at the carbohydrate concentration of those rhizomes. So the reason we do that is because the rhizome is the storage organ. And if we're interested in knowing if they're going to be viable, we wanted to see how much carbohydrate was in there, what ability does it have to actually resprout and put new tissues out. So that carbohydrate concentration was a measure of that viability. So I'm going to show some results here. This is first going to be biomass data. We're going to be looking at below ground biomass. And first thing that I want to point out is that the total submergence treatment mostly killed the plants. When we put these plants into water, completely submerging them, they did not survive. And that may be unsurprising. We drowned the plants died because they were drowned. However, I was a little surprised that there were basically none of them that continued growing after we put them in there to break the plane of that water. And you see that there's basically no new below ground biomass produced. In the cut to drown treatments, so the partial submerged that received cutting, we see then that the early cut was actually more effective than the late in decreasing the biomass productivity. And you see that the biomass was really similar to that of the total submergence when we applied that early cut. The late cut also significantly decreased it from no cut at all, but it was more effective early on. And then you can kind of see how cutting itself without submergence affected below ground biomass as well, not significantly. Now I'm going to show you data on these carbohydrate analyses. And again, the question here is, are these rhizomes viable? So I'm going to look at 3 different fractions. We have percent sugar, percent starch, and then a total structural carbohydrates. And really, the first one is going to be the readily available carbohydrate, the stuff that's ready to produce tissues now. The percent starch would be things that are more like longer term storage. And then the last one is the sugar and starch together. So first, again, when we look at those totally submerged plants that appear dead, we also see that those rhizomes have very, very low carbohydrate concentrations in all of the fractions. So they're very, we assume that those are likely inviable because they basically have no resources to be able to put into new shoots. Additionally, when we had a partial submergence and cut, both of those cutting timings produced similar results in terms of the low amount of carbohydrate concentration present in the rhizome tissues. So again, likely inviable because they do not have the resources to create new shoots. And then you can get a sense for how non-flooded plants were affected by cutting as well. In addition, we have also saved a couple of these tanks that were not used in that experiment, but had similar treatments applied to be able to get at this viability question in another way. So we've taken these plants and we allowed them to senesce naturally outdoors, and then we overwintered them in the fridge. And this year, at the start of this growing season, we're going to put them in our growth chamber and see the percentage of those that re-sprout. So we can get a sense of viability that way as well. Okay, so we saw really good success in the greenhouse on a really small scale, but the point of this is to understand the effectiveness on larger scale in the field. So I'm going to talk about a couple of field plans that we have, but first I just want to quickly go over a pilot study that we did last summer at the Detroit River International Wildlife Refuge, where we worked in an impounded wetland. So this was an area that was not currently flooded, but had the ability to be flooded. And so you can see me out here in the middle of this site looking at this Phragmites patch that was a manageable size for us to be able to cut, and the managers at the refuge had the ability to flood it. So we took the opportunity to go out and cut it down. I don't know if you can hear that, but yeah, it's me cutting.
Just
so
2 minutes, Wes. Sorry to interrupt.
Okay. So I took a lot of, so we cut the biomass down, and then bagged it up, took it out, and cleared the Phragmites off of the site. And then afterwards, then they flooded this site. They actually flooded a little bit later than we expected, and the Phragmites came back. So we went back out and cut under the water table at that time. And what's cool here is to see that the biologist went out and took a look here, and she points out maybe 10 Phragmites stems that were still present. So it was fairly effective in reducing Phragmites cover. We have plans then for this summer to really expand on what we did in the greenhouse and to look at sites across the basin where we can cut patches of Phragmites. And really what we're trying to get at is to find sites, both patches that are isolated, those that are completely in high water, and those that may be connected to shoreline Phragmites, to see what the effect of those connected patches and the ability for those low water zones to kind of save the high water zones is. And so we'll be testing early and late cuts in the field, and also looking at responses of biomass, cover, carbohydrate concentration, and reemergence in year 2. So here's a couple of locations where we're thinking about doing that work. And then we also want to get at this, this question in greenhouse as well about if these connections to non-submerged plants can impact the ability to cut to drown. So in summary, our experimental cuttings in the greenhouse were extremely effective. It seemed that the early cutting was modestly more effective, and that the rhizomes appeared to be inviolable after drowning, but there's still some further study needed on that to see what happens this year. We're working on the bulk of this experiment is really on the field implementation and experimentation. So over the next 2 years, we hope to learn a lot more about how this works out in the field, and how successful it may be, and the opportunity to do that with high water. So again, I really want to thank you for listening, and give a shout out to the team that worked on this project. Spencer Whidden was instrumental in all the work he did. He kind of led this effort. Also want to recognize our partners who are helping us find sites and are working together with us on this work. You can contact me, the email address below. And if I have time for any questions, I can take them now. And again, thanks for the opportunity to speak.